Hi, everyone. Welcome to episode 10 of Terpenes in Testing, the podcast. This broadcast is brought to you by Mace Media Group and Terpenes in Testing magazine, the industry's first publication dedicated to cannabis science. I'm Jason Lupoi, the editor-in-chief of Terpenes in Testing magazine, and I'll be your host as we navigate across the landscape of cannabis and psychedelic science. There once was a time when no one within the United States could study cannabis with the exception of the University of Mississippi. The sum effort of those studies did not consider cannabis as an option for diverse medical conditions, and despite the Department of Health and Human Services having patented cannabinoids as neuroprotectants, the plant and its THC still sadly remains on Schedule 1. With researchers increasingly able to evaluate the plant and the molecules therein, however, we are learning more each day regarding what the polypharmacy within cannabis can and can't do. So for this episode, I wanted to speak with Dr. Andrea Small-Howard. Dr. Small-Howard has 25 years of scientific research and executive experience in the biopharmaceutical industry, supervising research and development, manufacturing, and quality control divisions in the United States and China. She has been named an inventor on 60-plus patent applications. She envisages bringing more phytomedicines to the prescription drug market through an innovative AI-based drug discovery engine and biopharmaceutical drug development program for disease-specific indications. Dr. Small-Howard has researched cannabinoids for over 20 years and has published numerous studies. Dr. Small-Howard is currently the President, Chief Science Officer, and member of the Board of Directors at both GB Sciences Incorporated and their wholly owned Canadian subsidiary, GBS Global Biopharma Incorporated. She is the architect of a strategic vision at GB Sciences and GBS Global Biopharma to make safe, effective, standardized cannabinoid medicines available to patients where its use can be supported with rigorous evidence. To achieve standardization in their cannabinoid-containing therapeutic mixtures, the company is using synthetic cannabinoids produced under current good manufacturing practices, which are homologous to the plant cannabinoids. Hello, Dr. Small-Howard. Welcome to the show.
Hi, Jason. Thank you so much for inviting me to talk with you today.
Well, I'm eager to dive in. I've always been interested in the work that you've been doing. Last we spoke, you were doing some research on proving out the Entourage Effect. And I thought I would start there since that's kind of where we picked up last during a Mace Media webinar. And I was curious what the outcome of that work was and if you found the results surprising at all.
So it's good to start at the beginning, right? It's interesting because I would classify all of our research projects as related to how can you get the most efficacious mixtures out of the plants, but in a simplified version such that you can manufacture it under CGMP. And so that's kind of the challenge that we're always up against, right? Is you start off with plant extracts, and these plant extracts have great capacity. But then you need to figure out what to keep and what to get rid of in order to come up with a simplified formulation that we then patent and we bring into production, ultimately for the prescription drug market. And so the fact that one of the things that we're happy to report is that in all of the areas where we have looked to determine whether or not there would be an entourage effect, we have found it through a lot of trial and error through using cell and animal models. But the very basis for how we're able to obtain patents using molecules that are in a plant is that we're able to show that very specific ratioed combinations provide increased efficacy relative to the sum of the efficacy of the ingredients, meaning there's synergy there. And so not only is that something that we're interested in, it's been the way that we're able to have our patents issued. In fact, some of the USPTO has put in the letters where they've announced that we are having these patents issued to us, they say it's because we are able to demonstrate the enhanced effectiveness of the mixture that your grant get this patent. And so it's the entourage effect has been very good to us because it's how we're able to distinguish our mixtures from others And it's the basis for why we believe these are going to be incredible drugs in the future, because they do have enhanced effectiveness relative to using the single ingredients as drugs.
Well, that's exciting work, because Dr. Russo's Taming THC paper kind of postulated a lot of this, and I think it was not a stretch of the imagination whatsoever. There was definitely reports of anecdotal evidence of people saying, I feel better if I have a whole plant extract versus something in isolation. And I think that's really amazing, that you guys were able to prove some of this theory so quickly. And I was curious if that research covered just cannabinoids, or if I remember correctly, you were also looking at terpene synergies.
Definitely, yes. And so, so I guess one of the things that was surprising to us in the beginning is that we originally felt that the world was going to be broken down into the major cannabinoids and the minor cannabinoids and the terpenes. And we assumed, because that's what sort of the literature was suggesting, that it would be the major cannabinoids that were the key ingredients, and that the minor cannabinoids and the terpenes would somehow be lesser players. But that turned out to not be the case. The mixtures that were identified as being the optimal in terms of their effectiveness differed depending upon the condition that we were studying. So, for example, Parkinson's disease, which is our lead indication, they're the most effective mixtures did turn out to be based primarily on the cannabinoids. However, when you look at our chronic pain therapeutic program, there is the bulk of the activity from the original plant extract was found in the terpene fraction. And we were able to demonstrate at the cell level, and we're now continuing that work at the animal level, that there were cooperative interactions between terpenes. And so that was surprising to us, because like I said, we assumed that, like everybody else in the world, that the major players in these mixtures were going to be THC and CBD. But what we found is that for a lot of these different indications, there is no THC and there is no CBD in a lot of these things that we're actually working on. But we did not know that until we sat down to decide. We did a lot of rigorous testing to figure out which were the ones that were the most effective in the simplified mixtures. So we're looking for mixtures that include, say, between 3 to 5 ingredients is kind of the sweet spot as far as what we've been trying to determine, so that we can take that enhanced effectiveness from having mixtures, but have that simplicity so that we can manufacture it at scale.
Are the products basically that result from the studies, are they still predominantly cannabinoids with a little bit of terpenes added in, or are there much more elevated terpenes, like with your chronic pain model, for example? With
the chronic pain model, there are 2 versions that have no cannabinoids in it.
Oh, okay, solely terpenes.
Yeah, and that does not blow your mind. It blew my mind when we first started getting these results. But we took it further. So while some of the original testing that we used for discovery was looking at the ability for compounds to have an effect, we then spend a lot of time trying to characterize the mechanism of action, meaning, okay, we've determined that these compounds seem to work together. How do they do it? And so we spent some time doing both lab-based work and in silico work, where we were able to demonstrate the different receptors and the different pockets inside the receptors that were the active sites for not just the cannabinoids, but also the terpenes, so that we can elegantly describe the mechanism of action for these mixtures. Because just because you have more than one active ingredient, your standard pharmaceutical company assumes that you cannot describe the mechanism of action adequately. I'd say it's more complicated, but I think it's not impossible, and that's something that we're in the process of trying to prove.
So when you in the bio, I read that you're using synthetic cannabinoids, and that was one of my questions for you. If you were using synthetic cannabinoids versus ones extracted from cannabis, is that just based off of you're needing very high purities and not a lot of the other extraneous stuff that might come along through an extraction?
Yeah, there are a lot of reasons to move towards the synthetic homologs. And so one of the things I do want to stress is that while we're using synthetic cannabinoids, we're using homologs, meaning they're identical in their structure to the ones that come out of the plant. So it's really an alternate sourcing. It's not an alternate molecule, because there are other people who study synthetic cannabinoids that are analogs, meaning the structure doesn't look like anything that exists in nature. And so those ones, sometimes there's some bad effects of. So I just wanted to stress that, yes, we're using the synthetic versions, but the ones that are identical in their structure to the ones that come from the plant. But that was not always the case. When we were doing our discovery work, we were using whole plant extracts. And so we start off in our discovery process doing sort of a natural products chemistry approach. But then once we've got some good leads on mixtures that seem to be working, we then switch over to use the synthetic versions, partly because when I have to have material shipped all over the globe, and that's true, we're working with universities and companies in 9 different countries. And when you have to ship plant-based materials, the paperwork on that is insane. Believe it or not, having a synthetic version of a cannabinoid is very helpful when it comes to logistics of shipping the materials around. We also believe that when we take our materials to the FDA, that they will look favorably upon the fact that these molecules are manufactured under CGMP, which is to their standard. And so while I'm not saying that plant-based cannabinoids are less effective, I'm simply saying that for supply chain, logistics, and regulatory reasons, we switched to using the synthetic versions. The other thing that is also true is that for those rare, so some of the things that we're using in our combinations include things like CBD, for which there is a glut of. But we're also using some of the less common cannabinoids, and those, it's harder to get the quantities that you need from the plant. So that's also a factor.
Right. And is that the case for the terpenes as well? Or are they, synthetically derived, or are those from, botanically derived?
At this point, the terpenes that we're using are still botanically derived, because terpenes form the basis of the cosmetic and fragrance industry, and because they're used so widely as flavorings in foodstuffs. You can, in fact, find very pure forms of the botanically derived terpenes. And so at this point, we're using the botanically derived ones, but that doesn't mean that we will necessarily do so. We're looking into seeing whether there's any advantage to having fully synthetic terpenes or not. Right now, it doesn't seem like it, but you never know.
Okay, that makes sense. Can you discuss how you developed your cannabinoid formulation for Parkinson's disease?
Sure. So it started with an observation. So prior to joining GB Sciences, I was working for a biotech company down in La Jolla, California. And that company was working on a therapeutic program for Parkinson's disease. And at that time, I was actually their business development person. So I was talking to a lot of folks about whether they wanted to help us monetarily with this research. And we established quite a good rapport. And every time I talk to them, we do always talk about, how are they going? How are things going? How are their symptoms? And they were always sharing with me the different programs that they were using to try to manage their symptoms. And even though this was in California, and medical cannabis was fully legal at the time, there started to be a trend. Patients were telling me that they were trying medical cannabis, but in sort of a hush-hush voice, even though it was legal, and that it was helping their symptoms better than anything else. And these patients were doing things like deep brain stimulation. They were going down to Mexico to get stem cell replacements. They were really pushing the envelope as far as the extremes of treatments that were available for Parkinson's. And there seemed to be a consensus that the cannabis-based formulations were working the best for them, despite the fact that there really was no regulation in the materials that they were receiving. Meaning some people were taking edibles, some people were using different delivery formats, and none of them really knew exactly what the active ingredients were using. They just knew that it was working. So some people might have just dismissed this as being sort of the placebo effect, but it happened so frequently that I thought there's got to be something to it. And now you'll see on YouTube, there's all these different testimonial videos of patients with Parkinson's that are having severe motor symptoms, and they'll consume cannabis in different forms, and you'll see that they're seeming to have symptomatic relief. And so, I became a real believer that there had to be something in the plant that was causing these effects. And so through a series of experiments where we were using in silico machine learning tools to try to figure out which of the compounds in cannabis would potentially be helpful for helping with the motor symptoms, we then took it into cell and animal work, and we've derived at these formulations that have 3 ingredients. So there's 3 different formulas that have 3 ingredients each that have shown to be able to reduce the motor symptoms of Parkinson's in an animal model. And those are the ones that we're now bringing forward into the clinical trials. And so it's been a long process of, like I said, sort of trial and error. They call it screening, but we're feeling fairly confident going into our first in man that we should be able to help people in a very reproducible way, right? Because you don't have to rely on what happens to be in your local dispensary. We've identified compounds that in very specific ratios that should work for you every single time you take it.
Right. That's always kind of a key thing, because nobody's really given the chemistry or a breakdown of the chemistry in a given product when they're at the dispensary. In Pennsylvania, we require terpenes to be on all products, but I've noticed that it's not always the same terpene list, and you don't see that prior to shopping. So you may buy a product and then see the label. It's not on product menus. And if you're trying to replicate a product that was effective for you, it could be quite difficult to do so. So that reproducibility, I think, especially when we're talking about something as debilitating as Parkinson's disease, seems paramount to success in the treatment.
Absolutely. The other thing that we've noted in our work is that the more complicated the mixture that you're trying to create, the less likely that you'll be able to get the same effectiveness every time. And so that's the other reason to reduce the number of compounds that are in there, is that we've gone through a lot of sequential testing that shows that, okay, if I take this particular cannabinoid, add this other cannabinoid to it, and then I get a certain result, and then I add that third one to it, I get this different result. When I get to the 4th and 5th one, it starts to be harder and harder to predict which way it's going to go. So 3 seems to be one of the sweet spot of where you get molecular cooperation, but you don't start to step on some of the other positive synergies. Does that make sense?
Yeah, absolutely. That makes a lot of sense. I was curious, just for our listeners who may not be familiar with the concept of machine learning, could you briefly just touch on how that works and how that kind of kickstarts the process?
Sure. So one of the things that we're in the information age, right? And one of the things that is true is that when people used to be doing drug discovery work, they didn't really have access to huge amounts of data or huge computer power. And in this day and age, however, we have that at our disposal. We have big data sets, and we have machine learning tools that will allow you to look for patterns in these large data sets that are very difficult to do by eye. And so when I say machine learning, that's kind of what I mean is the ability to let the computer take over and look for patterns that becomes overwhelming because of just the sheer amounts of data. The other reason that we call it machine learning is that it's iterative, meaning you don't just do this analysis once. You keep feeding it data, and over time it gets better and better at its predictions. And so we've been working with our AI-enabled drug discovery engine now for almost 8 years, and we've been training it with all the data that we have. And so every time it makes a prediction, it's drawing from not only enhanced data, but it also has experience in running those kinds of patterns. So it gets better and better at its job. Is that helpful? So,
yes.
So a lot of times, so sometimes it can be as simple as we're asking it to analyze pre-existing databases. We work a lot with traditional medical systems. We've taken several of the major traditional medical systems in the world, and we put it into a database where all of the data exists in a single interrogatable space. And that allows us to ask questions broadly about, if you're going to use plants for medicine, how has it been done in the past and how has it been working? And so that's another aspect of how we use the AI and machine learning is in these, what we call in silico convergence analysis, where we basically say, for example, we have designed some novel pain therapeutics, going to the database, and we say, all right, these traditional medical systems have been using plant-based compounds for thousands of years in very different parts of the world. If you look at the subset of those ingredients that you can find in common between these things that have evolved independently, that sweet spot of the things that they have in common shouldn't be there by accident, right? It should indicate that these compounds are probably really important in the treatment of pain because of the thousands of years of trial and error that have gone into developing traditional medical systems. And so when you do that analysis, one of the classes of molecules that turns out to be incredibly important are the terpenes.
That's super exciting, and it's nice that you have that tool available so that you can screen through a lot more molecules than what would otherwise be possible. I wanted to switch gears. It seems like very few conversations these days do not mention COVID, and I wanted to talk a bit about COVID because there's been some interesting studies regarding the validity of cannabinoids and terpenes from cannabis being efficacious against coronaviruses in general, like COVID-19. And I think that that's a really interesting and noteworthy point here because this was a virus that the world had not seen, and yet this ancient plant still has some tricks up its sleeve for providing some kind of relief in different regards for COVID. And I saw that GB scientists had filed a patent for a proprietary formulation for treating acute respiratory distress syndrome, which is the leading cause of hospitalization and deaths. And so I was curious what you could tell us about cannabinoids and COVID and how that formulation came about.
Well, yeah, thank you. Though this is a really important topic, I think, and it's interesting because we're obviously not the only people that are doing research in this area, and the research in this area kind of falls out into two different categories. There are some people that have been demonstrating that cannabinoids and terpenes and mixtures of cannabinoids and terpenes can physically, mechanically block the infection of your cells by adhering to the spike proteins on the outside of the COVID-19 the virus, right? And so that's very interesting and important results. However, one of the things that we've noted about these coronaviruses and viruses in general is that they mutate quickly. So this kind of strategy where you have an interaction directly with the virus is often limited because the part of the way that viruses evolve is they change their surface proteins so that they can evade immune responses, right? So we took the other tact, and there are other companies that are doing this as well, where instead of trying to find molecules that directly block the infection of COVID, what we're doing is we're trying to protect patients from the secondary effects of having a viral infection, right? And so what we've done is we started with some work that we were doing at Michigan State University. And at the time, what we were doing is we were looking to help those patients who are living with HIV, because I don't know, not many people know this, but people who are living with HIV and who are taking their meds and are have undetectable serum levels of HIV, almost all of them still have a population of the HIV virus that lives in their brain. Because there's something called a blood-brain barrier, viruses can hide in your brain where the immune system doesn't have the ability to go in and clear them. And so what the researchers that we were working with at Michigan State University had noted, and others have noted too, is that this brain resident population of HIV was causing inflammation. And these secondary to this inflammation, patients were having what they call HAND, or HIV-associated neurodeficiency syndromes, which present somewhere like Parkinson's, but also a little bit like Alzheimer's. So one of the things that has been hypothesized is that a lot of the neurodegenerative diseases seem to have an inflammatory root. And so what we originally were doing was we were simulating a viral infection, and we were trying to see which of the molecules from the cannabis plant would be helpful in trying to downregulate that inflammation. because we were trying to develop this program to help patients with HAND, or HIV-associated neurodeficiency syndrome. But then right after we had collected a bunch of data on how cannabinoids and terpenes both can very precisely target aspects of the immune system, we were into COVID, right? And so I remember thinking at the time, we have all this data on how the compounds from the cannabis plant can be used to regulate the immune system in response to a viral infection. There ought to be something in here that will be relevant. And sure enough, when we started looking, there was. What we've done is we've formulated some mixtures that are, what their goal is to do is to leave those parts of your immune system that fight viruses intact, because you want to be able to fight not only COVID, but also when you're in the hospital, especially if those people are hospitalized, the hospitals are full of viruses that are pretty nasty, right? So ideally what you want to do is keep the part of your immune system going that is going to be helping you fight viruses, but you want to suppress the parts of the immune system that are causing damage. And there's a phenomenon called hyperinflammation, or cytokine release syndrome, or cytokine storm. These are all names that have sort of a similar meaning. But in COVID and some of the other viruses that are very damaging, it's not necessarily the virus itself that causes the greatest damage. It's the overaction of your own immune system starts to damage your own tissues. Specifically, it goes for the lungs and the heart, and these are key organs for us, right? So we said, okay, if we can regulate the immune system such that we suppress the cytokine storm, then and leave your immune system such that you can still fight viruses. This is pretty much an ideal treatment strategy for not just COVID, but for any of these viruses that cause hyperinflammation like that. And so that is the course that we're pursuing, and we're very hopeful for its utility, not just now, but in the future.
So that's really encouraging as well, and I think that our conversation has revolved around Parkinson's and pain and coronavirus, and I'm curious, for my last question, I'm curious, given that your research has demonstrating the effectiveness of cannabinoids for a range of different conditions, I was curious what your thoughts are on what this says about the endocannabinoid system and its importance in our bodies, and also what it says about the fact that it's still not really being taught to medical professionals.
So I'm going to start with the end of your question and work backwards. So I think the endocannabinoid system should be a required part of every medical professional's educational upbringing. I noticed that there are a few CMEs now that will cover the endocannabinoid system, but it's not necessarily being adapted into the main curriculum for a lot of medical schools, and I think that should definitely change. The other thing I'd like to say is that it is a viable target for doing drug development work. That is what our work seems to be demonstrating. One thing I do want to point out, though, is that this research is not for the faint of heart, because what most people who are doing drug development work do is they say, all right, I'm going to be looking for a single ingredient to hit a single receptor. But the endocannabinoid system is full of receptors and molecules that are working together, and there are other receptors that are now when they first described the endocannabinoid system, they basically were talking about CB1 and CB2, or cannabinoid receptor 1 and cannabinoid receptor 2. And they were talking about the fact that in response to stimuli, the cells of our body are able to synthesize with what are known as the endocannabinoids on cue. So when there's a trigger, we synthesize an endocannabinoid and it hits its receptor, and you have almost instantaneous loops, right? But what that is, that quickness, I think, that made it so that we didn't see that system for a long time. It took a while. It wasn't until we started playing with the plant-based cannabinoids, the phytocannabinoids, and the synthetic cannabinoids, the ones that don't look like the plant-based cannabinoids, that we were really able to sort of freeze time and show that the system was there. And in recent years, we're finding it's not just CB1 and CB2 that are sensitive to cannabinoids and terpenes. there's a whole family of receptors called the TRP channels that are sensitive to cannabinoids and terpenes. There are things like most of your neuropeptides or the neurotransmitter receptors. Those are also often sensitive to cannabinoids and terpenes. So we have to redefine what we mean by endocannabinoid system a lot as we're learning more and more of how vastly it affects the systems in our body in a coordinated way, which is another reason why it's good that we live in an information age. It's good that we're in an age where we can put a lot of this data about the parts of the system and all of the complex data sets into a computer and ask for it to help us in learning about the patterns of how to make these systems work in our favor as far as making new medicines.
Absolutely. And Dr. Small-Howard, I want to thank you for discussing the work that you and your colleagues and collaborators are endeavoring, and we're definitely keen to hear more as you continue to demonstrate the effectiveness of cannabinoid medicine.
Thank you very much.
Thanks all of you for tuning in. We'll see you next time on Terpenes and Testing, the podcast. Until then, please come visit us at www.terpenesandtesting.com or on one of our social media platforms. Cheers.